Students, today we are going to discuss about the cell cycle and cell division. After getting some idea regarding to the cell, which is the basic structural and functional unit of all the living organism, we are going to learn about what is the cell cycle and cell division. Listen, the process by which new cells are formed from the pre-existing cells is called cell division. Eukaryotic cells show highly regulated, systematic and controlled cell division called as mitosis and meiosis. These are the two types of cell division. Mitosis is an equational division. It is the characteristic division of somatic cell. That means your vegetative, or you can say it as body cells. Meiosis is the reduction division. It is the characteristic division of reproductive cells. Look at here. This is the graphical representation of your cell cycle. Generally, the cell has three main phases. The cell cycle has three main phases. One is interphase, mitosis, cytokinesis. These all things occurs one after one in cyclic manner. Okay. During mitosis, the nuclear content of the cell divides. During cytosis, the cytoplasmic content of the cell divides. And during G1, the cellular content excluding the chromosomes are duplicated, like your centrosome and other organelles. Then in S phase, it is called as synthesis phase. Each of the 46 chromosomes is duplicated by the cell in case of human being. In G2 phase, the cell double checks the duplicated chromosomes for error, making any needed repairs. If the cell has not duplicated all its chromosomal content, then the cell undergoes another stage which is called as G0 stage or cell cycle arrest stage. You can see here. Look at here. You can classify the whole cell cycle into two main parts. One is interphase, another one is cell division, or particularly M phase. That may be your mitosis or meiosis. And during interphase, the cell undergoes three main phases. One is G1 phase, then S phase and G2 phase. Then the cell division again are of two types. One is karyokinesis. That means during that phase, the nucleic acid content, or especially the chromosome, doubles and divides. Again, it is of four main stages. One is prophase, then metaphase, anaphase, and final stage is your telophase. Whereas cytokinesis occurs after the nucleic acid content division or your chromosomal content division. During cytokinesis, the cytoplasmic content separates from the mother cell. Okay? One thing you have to remember, when the cell undergoes cell division, the chromatin fibers condenses into chromosomes. And when karyokinesis started, at that time, the nuclear membrane gradually degenerates. Clear? Look at here. And G0 stage is the, look at here, that G0 stage is the non-proliferative stage, or it is called as quiescence phase. Okay, that means during that period, if cell undergoes any error, the cell undergoes some rest stage. Okay, and again the cell has two checkpoints in G1 and G2 respectively. Okay, if the condition is not suitable for the cell division, then the cell cycle may be stopped or it undergoes G0 stage to retain its complete maturity. Then another complex is there which controls the progression of cell cycle. That complex is called as CDK complex. Okay, CDK stands for cyclin dependent kinase. Look at here, your CDK stands for cyclin dependent kinase, which is one type of protein breaking enzyme. Okay, and it depends upon cyclin. So cyclin is one subunit and that kinase enzyme is another subunit. Okay, they both forms cyclin CDK complex. They both forms what? Your cyclin CDK complex. Look at here, we are going to discuss about first type of cell division that occurs in your somatic cell. It is known as your mitosis. Otherwise it is called as equational division, because during this division each daughter cell receives the same number of chromosomes and same characters as that of the original cell. Here daughter cell means the new cells that forms from the original mother cell, and it has four main stages that we have already discussed. One is prophase. Look at here, this one is prophase, metaphase, anaphase, telophase. Then after up to this much your cell undergoes the division of your nucleus. Then after the cytoplasmic content divides when the cell enters cytokinesis stage. Clear? And another special stage here you can see that occurs before metaphase is termed as prometaphase. Okay, so let us have a discussion about each and every stages of mitosis. Look at here, during mitosis the first stage, that means in prophase, the nuclear membrane and nucleolus disappears. They start to disappear and chromosomes become shorter, thicker and prominent. The chromosomes remain scattered in the nucleoplasm region at the end of prophase. You can see it there. Okay, look at here, all the diagrams has been depicted the various changes in the cell. Okay, then in prometaphase what happens, you can see that the nuclear membrane has completely disappeared. Okay, and from the centrosome the spindle, it is called as the spindle pole, the spindle fibers already started to elongate and the polar microtubules are joining with the chromosome. You can see some nuclear envelope fragments are also present inside the cell cytoplasm. The kinetochore microtubules generally they connect to the centromere. Okay, generally you can see here there are two types of microtubule. One microtubule is your polar microtubule that connects from this pole to this pole of the cell. These poles are called as spindle pole. Okay, and another one is your kinetochore microtubule, that means it connects to the kinetochore present at the two sides. During prophase 1, what happens? Look, during prophase 2, the chromosomes gradually become visible and distinct. The nuclear membrane and nucleolus disappear. At the end of prophase 2, each chromosome has one centromere and two sister chromatids. Then next is your metaphase 2. You can see during metaphase 2 what happens. You can see the organization of bipolar spindle body occurs. That means you can see that these spindle body, these are the spindle body, they are remaining at the polar end. Arrangement of the next point, arrangement of the chromosomes along the equatorial plane occurs. Metaphase means they have to come to the middle plane or equatorial plane or equator of the cell. Then two sister chromatids of each chromosome, they face to opposite each other. Centromere of each chromosome get connect with the chromosomal fibers or your microtubules with the pole. Then next is your anaphase. During anaphase what happens? They are coming away. Look at here, the chromatids, they are coming away from each other. Okay, you can see they are going away from each other, sorry. Then centromere of each chromosome divides longitudinally into two centromeres. As a result, each chromosome divides into two chromatids, that is daughter chromosomes. The sets of daughter chromosomes move towards opposite poles. Next is your telophase 2, and during that phase what happens? The chromosomes, they again returns to the opposite end, that means terminal end. Okay, so here 4 nuclei are formed. Each nucleus is haploid and hence different from the original diploid nucleus. That means the reduction in their ploidy level occurs, that means chromosomal content occurs. The 4 daughter nuclei differ from each other in details. Okay, though they have 4 daughter nuclei, but they are completely different because of the crossing over. Remember that point. Okay, during crossing over what happens? We have discovered that recombination occurs, that means recombinant chromatids has formed. Then after your telophase 2, the cell undergoes cytokinesis, and again cytokinesis are of two types. Wait, I'll show you. Look at here. What are these two types? One is simultaneous type, another one is successive type. What is simultaneous type? Simultaneous type means you can see that here the cytokinesis, that means cytoplasmic division, occurs only once after the whole division of or separation of your nuclei. Then but look at here initially the diploid area, there was your diploid cell. Then after meiosis 1 or reduction division, from here you can see that 2 haploid nuclei has formed. That means that cell remains intact, but 2 nuclei has formed. Then after inside that nucleus you can see again that from one nucleus now you have 4 nuclei. Then after the karyokinesis occurs, cytokinesis occurs. That means now the cytoplasm splits and forms 4 daughter cells. Okay, but in case of successive type of division, the cytokinesis occur twice. What happens at that case? The initial diploid mother cell after meiosis produces two haploid daughter cells, okay, by splitting the cytoplasm. You can see. Then after, again one cell splits into its next two daughter cell by again splitting its cytoplasm. So here you can see that the cytoplasm splitting occurs two times, so that it after each division of nucleus, so that this type of cytokinesis is called as successive type of cytokinesis. Clear? Now look at here. So overall, what we have seen that the nuclear division occurs twice. That means in M1 and M2. Nuclear division means the chromosomal contents. The nucleus divides. Then the chromosome divide once. When? During meiosis II during the meiosis II, at which stage? At the anaphase 2 stage or double I stage or second stage. Then separation of homologous chromosome occurs only once. When? At the anaphase 1 of meiosis I. Again, the cytokinesis may occur once when the cell undergoes simultaneous type of cytokinesis, and that cytokinesis may occur twice when it undergoes successive type of cytokinesis. Then the crossing over occurs only once. When? Now at the Pachytene stage of prophase 1 of meiosis I. You can see that diagram. Then it is very much important point. Remember, when the crossing over occurs at the Pachytene of prophase 1 of meiosis I, that causes the formation of recombinant chromatids or exchange of characters. Exchange of chromatid means exchange of DNA. Exchange of DNA means exchange of gene. Exchange of gene means exchange of character. Okay. Then that causes variation, remember. Then the reduction of your diploid chromosome occurs only once. When? At the anaphase 1 of your meiosis I. Remember, anaphase, when the chromosomes are separating from each other by going away from each other at the meiosis I stage, which was actually the reduction division. Now, what are the importance of your meiosis? Meiosis is essential in sexual life cycles to regulate the number of chromosomes in species. Meiosis also plays important role in organic evolution through genetic recombination due to crossing over. Okay. Then what is the importance of that kinetochore? Kinetochore, as you know that kinetochore remains around the centromere region. If you take this one as the chromosome, this constricted part is called as centromere, and around your centromere you have the kinetochore, which connects with the spindle fiber or your microtubule with the polar region. So this one is the importance of your kinetochore. It causes the separation of allele, allele means your paired chromosome, and reduction in the number of chromosomes. And that occurs in meiosis I. And during meiosis II, it causes the division of chromosomes. Okay, that we have already seen the anaphase 2 of meiosis II. Now comes towards the point of discussion that the meiosis again are of three types. One is zygotic meiosis, next one is sporic meiosis, the final one is gametic meiosis. Look at here. The zygotic meiosis occurs in the diploid. Look at here. It occurs in the diploid zygotic nucleus that forms after fertilization, or you can call it a synapsis, syngamy. Syngamy means fertilization or fusion of gamete. So that your meiosis occurs when it occurs in the diploid zygotic nucleus in case of most algae and fungi. Since it occurs in zygotic stage, such type of meiosis is called as zygotic stage. Okay, then the sporic meiosis occurs in the diploid spore mother cell during the spore formation. It occurs in the diploid spore mother cell of the spore forming organisms. What are they? They are the bryophytes, then your pteridophytes, then your gymnosperms, and another group which can create spore, they are the angiosperm. Okay, these four spore forming plants in their diploid spore mother cell performs sporic meiosis to produce spore. Then last one is your gametic meiosis, and it occurs in case of our highly evolved animals. That means those who are able to produce gamete from their diploid gamete mother cells. Okay, look at here. You can see that the zygotic meiosis occurs in algae and fungi in their zygotic cell, and your sporic meiosis occurs in the spore mother cell of your spore forming plants, generally your bryophytes, pteridophytes, gymnosperm and angiosperms, and gametic meiosis occurs in your gamete forming organism cells, especially your gamete mother cells, which are present in your germ epithelium of your reproductive organs. During the formation of gamete, that gametic meiosis occurs in the higher developed animals, most of the animals, in all animals. Okay, those who are reproduces sexually by forming the gamete. Then some important events you should remember for your entrance point of view. In plant cells, the mitosis was first described by Strasburger in 1875. In animal cells, the mitosis was first described by W. Fleming in 1879. Then the term mitosis was coined by W. Fleming in 1882. The term meiosis was coined by your J.B. Farmer in 1905. Thank you. Have a nice day. If you will see the structure of one chromosome, I want to show you. Look at here. You can see that constricted part is called as centromere, and around that part you have your kinetochore. Okay, and when that kinetochore joins with the spindle pole by the microtubule, that type of microtubule are called as kinetochore microtubule. Okay, this type of microtubule. Now come towards the metaphase. Before going towards the metaphase, you have to learn about at the middle place of one cell you have one plane. That plane is called as your equatorial plane. Okay, and gradually when scattered chromosomes get attached with the spindle pole by the microtubules align all these chromosomes to the equator of the cell. Then the nuclear envelope gradually start to disappear. Okay, and and this stage is called as metaphase. That means all the chromosomes comes to the middle part of the cell. Prophase means this one is the primitive phase under which the primary stage of cell division occurs, or initial stage of cell division occurs. Then after, during metaphase, the chromosomes comes towards the middle portion, that is your equator region of the cell. Then after, during anaphase, the chromosomes start to split from the centromere region, that means at the middle, and they are being pulled towards the pole. Okay, and as you know that, look at here, again I want to show the diagram. If this one represents your chromosome, these two parts are called as your arms of chromosome. Okay, and this part, look at here, this part is called as your chromatid. This is another chromatid. You can see that one part of chromatids are coming towards one pole, and that another part of chromatid of one chromosome coming towards the another pole by separating from the chromosome from the equator region. Clear? And that occurs because of shortening of kinetochore microtubule. Okay, then that causes separation of splitted chromosomes, that means your chromatids. Then in telophase, what happens? The cell reach at the terminal end of the cell, that means at the polar part of the cell. At that time, what happens? Microtubule completely shortens, and all the nuclear envelope start to reform around the individual chromosome groups. You can see there are two types, two groups has already been formed, and the nuclear envelope starts to reform around them. Okay, that's all about the telophase. Then that all the processes are called undergoes your karyokinesis, that means breakdown of nucleus, which is followed by cytokinesis, and then after, the original cell divides into two similar and uninucleate daughter cells. Okay, you can see here. Then the cell divides. Okay, that's all about your mitosis. What is its importance in unicellular organisms like Monera and Protista? Mitosis is a means of increasing the number of individuals in the population, that means reproduction. But in multicellular organism, it helps in growth of organism and repair of tissue. Okay, then now come towards the next type of cell division, that is your meiosis. Before going to learn about meiosis, I want to share some basic terminologies regarding to the cell division. Look at here. Here it represents the homologous pair. That means same size chromosomes are there. Let us consider this one is the chromosome A and this one is chromosome B. Here you can see that they are looking similar to each other, and as we have discussed, this is the arm of your chromosome, and this part is called as your chromatids. Okay, it is called as chromatids. I have to change the color. It is called as chromatid. it is also another chromatid. They both are joining with each other by the kinetochore. Okay, and this constricted part is called as centromere. Remember, and the chromatids of one chromosome is called as sister chromatids, whereas the chromatids of two different chromosomes are called as non-sister chromatids. Okay, and one chromosome of one homologous pair is called as dyad because it has two sister chromatids. It has how many sister chromatids? Two sister chromatids. Clear? So that it is called as dyad. Di means double or two. Look at here. Another term is there, tetrad. What are tetrads? Tetrads form during the metaphase 1. What is the metaphase 1? We will discuss much in later, but that occurs during meiosis. Simply remember right now, during meiosis, the tetrads form. A tetrad, or it is called as bivalent, is two duplicated homologous chromosomes. That we have already seen previously, that the homologous chromosomes are also called as dyads. One dyad, look at here, this is one dyad, and this one is another dyad. Okay, so a tetrad is formed by the joining of two dyads or homologous chromosomes. You can see here, this whole structure is called as your tetrad. The process of dyads pairing of across equator of the cell is called as synapsis. Okay, that means when homologous chromosome start to pair with each other, that means they start to align with each other by coming closer to each other. This process is called as synapsis, and this part is called as chiasma. Chiasma or chiasma you can pronounce. Okay, now look at here. Here you can see, and what is crossing over? Look at here, when homologous chromosomes align together, look at here, when they are coming closer to each other, at that time what happens? The two non-sister chromatid, look at here, this chromatid and this chromatid, they are the non-sister chromatids, and when these two non-sister chromatids comes closer to each other, they coil around each other. That forms the crossover. And during crossover, what happens? Some part of chromatids get exchanged with that non-sister chromatids. Look at here. Here this red part has exchanged from this part, and that blue part has exchanged from that blue chromosome. That exchange of chromatid phase causes recombination. Look at here. So that now we can say that these chromatids, this chromatid and this chromatid as the recombinant chromatid. Except these two chromatid, you have another, this chromatid and this chromatid. These are called as non-recombinant chromatids because they have not crossed over each other. That means crossing over has not occurred in between these two chromatids. And the chromatids in which the crossing over has occurred, you can see that there is the change in part of chromatids. Hence, those chromatids after crossing over are called as recombinant chromatids. I hope you all got the terminologies. Look at here. The meiosis is called as reduction division. It is a characteristic division of reproductive cells only. It doesn't occur in vegetative, that is your somatic cells. It takes place in diploid cells. Diploid cell means having two end set of chromosome that we have already discussed. Now, when a diploid mother cell divides by meiosis, it produces 4 haploid cells. That means carrying one set of chromosome. Since the number of chromosome is reduced from 2n to n, 2n to n, meiosis is described as the reduction division, whereas your mitosis was called as your equational division. Okay. Now, meiosis occurs in the life cycle of only those organisms which are sexually reproducing. Meiosis and fertilization are the two important and essential events in the sexual life cycle. They help to regulate and maintain the number of chromosomes through the alternation between diploid and haploid phase in their life cycle. You can see there is a alternation in generation in some lower category of plants. That means they have the diploid phase of division and haploid phase of division. Then karyokinesis in meiosis consists of two complete nuclear division called as karyokinesis, means the nuclear division, especially your chromosomal division, and it occurs in two division. That means two phase. One is meiosis 1 or M1, another one is meiosis 2. Actually, that meiosis 1 is reduction division, because during this process the separation of homologous chromosome occurs. That means the diploid mother's nucleus, look at here, the diploid mother nucleus reduced into the haploid daughter nuclei in, and that occurs in meiosis 1. In meiosis 2, it is similar to our mitosis previously, and during that division, what happens? The equational division of chromosome occurs. It involves the division of chromosome during during anaphase, that we have already discussed. There are 4 main stages of your cell division in mitosis: prophase, metaphase, anaphase, and telophase. That means during karyokinesis. So similarly, in meiosis 2, during anaphase, you can see that there is equational division of chromosome at its anaphase stage, because it is similar to your mitosis. Okay. Then the time interval between the chromosomal division, that means karyokinesis of M1 and karyokinesis of M2, is called as interkinesis. Okay, that means the gap present between meiosis 1 and your meiosis 2 is called as interkinesis. What was that? It is called as interkinesis. Okay. Now let us discuss about the meiosis. And look at here. After karyokinesis, that means nuclear division or chromosomal division, your cytokinesis occurs. That means cytoplasmic division occurs, like your mitosis. But in case of meiosis, that cytokinesis are of two types. But that's all about your latter part of your discussion. But first we have to discuss about the first part of your meiosis, that is the meiosis 1. Now let us have the discussion. Like your mitosis, meiosis 1 has its 4 stages. What are these? One is prophase, then metaphase. Listen, it is called as your prophase 1, prophase 2, prophase 1, metaphase 1, anaphase 1, telophase 1. But the prophase 1 is the lengthiest or the longest duration of your meiosis, all over the meiosis, because your prophase 1 again has 5 subdivisions, that means sub stages. What are these? One is your leptotin, then second one is zygotin, third one is pachitin, fourth one is diplotin, and the fifth one is your diakinesis. Okay, again repeat with me: leptotin, zygotin, pachitin, diplotin, diakinesis. Clear? After completion of these 5 substages of your prophase 1, we have to go the rest 3 phase: metaphase, anaphase, and telophase 1 of your meiosis 1. So overall these all stages have the similar stages like your mitosis, but one difference you can see at here, that the 2n number of chromosome here reduced into n number of chromosome or n set of chromosome. Okay, but in your meiosis 2, again I am repeating, what happens, that n number of chromosome will remain same as n number of chromosome as your mitosis or your equational division. So we have to discuss, we have to discuss much more clearly about various stages under your prophase 1. First is your leptotin. Look at here. During leptotin, what happens? Listen, the chromosomes visible as very long and thin threads during the leptotin. Okay, and still the nuclear membranes are present, because this is very primitive stage. Then next come to the second stage, that is your zygotin. During zygotin, what happens? The synapses, that means that you have already said previously, the pairing of homologous chromosomes occurs. Okay, and the paired chromosomes are called bivalents. Look at here. These homologous chromosomes are, now you can say them as homologous chromosome, or you can say them the bivalent. Okay, and each pair is in dyad stage. Look at here, this remains in dyad stage, that means each chromosome has its 2 chromatids. Okay, and each that dyad stage you can see during the second substage of your meiosis 1, that is your zygotin. Okay, during zygotin you have the dyad in homologous chromosome pair or bivalents, that means there are 2 chromosomes. Okay, pair chromosomes, so that they are called as bivalents, and each pair is in dyad stage, that means each pair has 2 chromatids. Okay, and then during pachitin, what happens? Each chromosome has 2 sister chromatids joined by 1 centromere. Hence, the bivalent or your homologous chromosome pair are in tetrad stage. Okay, don't be confused. In zygotin, the chromosomes remain in dyad stage, In Pachitin, the chromosomes remain in tetrad stage. That is, tetrad stage means there are 4 chromatids in each pair. Now you can imagine that all the homologous chromosomes, they have connected with each other by forming that means they are going to cross over each other, okay, by forming the synaptonemal complex. You can see here that okay, that synaptonemal complex will form in the next phase. Now during that stage you can see that such type of synaptonemal complex, that means they will cross over with each other to share the part of your chromatids. Okay, now look at here. Then next substage of your meiosis 1 is diplotin, and during diplotin what happens, the chiasma form. Chiasma means the junction in which you can see here that the crossing over has occurred by forming the synaptonemal complex, okay, and then after the repulsion begins in between the 2 homologous chromosomes. That means the terminalization process begins. That means they are again going to separate from each other by splitting the part of chromatids after recombination, okay. Then after the last substage of meiosis 1 is diakinesis. During diakinesis what happens, the pairs of homologous chromosome remain scattered again in nucleoplasm, and the nuclear membrane, you can see that nuclear membrane is gradually disappearing, and the nucleolus along with it also disappears, okay, start to disappear. Then after these all are the stages of your prophase 1. After prophase you have the metaphase 1, okay. So during metaphase 1 what happens, the pairs of homologous chromosome, you can see look at here, look at this diagram. The pairs of, you can see here that the crossing, after crossing over, the recombinant sister chromatids has already formed, okay. And during metaphase 1 what happens, the pairs of homologous chromosomes are arranged along the equator plane. As you know that during metaphase they have to arrange at the middle part of the cell, that means the equator part of the cell. In each pair the 2 homologous chromosomes are facing the opposite poles, and each chromosome is connected to the pole on its respective side only with the microtubules. Then after during anaphase, look at here, then your anaphase 1 stage. During anaphase what happens, the process of that terminalization, that means separation of chromosome is over. Then the 2 sets of chromosomes move towards the opposite pole when the microtubules attached to the kinetochore shortens, okay. When it becomes reduce its size, when it will reduce its size, then they will start to move towards the opposite pole by separating, or by after the completion of your terminalization from the equatorial plane, okay. Anaphase. Then telophase, you have to remember telophase similar to your tail, that means last, this is the last phase, okay. Anaphase, remember they appear to come to the opposite sides, okay. They are moving away. Anaphase, they are moving away from each other. Metaphase, they are living in the middle plane or equatorial plane. Anaphase, they are moving away from each other. Telophase, this is the terminal stage. Telophase, you can what relate it with some of the relevant words, okay. So during anaphase what happens, all the chromatids or the chromosomes, they are moving away from each other, and they start to migrate towards the polar region. Then in the telophase 1, under meiosis 1, what happens? See here, you can see that a nucleus is organized at each pole. Again the nucleus has organized, you can see, and each daughter nucleus contains only one set of chromosome. Hence meiosis 1 is reduction division. Clear? That's all about your meiosis 1. Look at here. Then we have to discuss about the meiosis 2, and it is the equational or similar to mitosis. It involves division of chromosomes during anaphase 2. We have already discussed in what happens in anaphase stage. That means the chromosomes moving away from each other. Anaphase moving away from each other. So similarly, what will be the stages of meiosis 2? Yes, you can see the first stage will be prophase 2, then metaphase 2, anaphase 2, telophase 2. Then after, your cytokinesis will occur. Okay.